Okay, we're going to go ahead and start now. This is the second half of Unit 1. We're going to take a look today at mitosis and cell division, and we're going to look at membrane transport, diffusion, osmosis a little bit, something we've talked about. Started this in lecture. This will be a little more information on it. The lab test will open up next Thursday, the 9th. It will stay open for 10 days, so it'll close on the 19th. It's going to be 50 questions. It will be multiple choice. It will be mostly identification. It'll all be multiple choice, but it will be mostly identification based on the slides from week 1 and some of the slides we see today, and questions that identify the microscope, identify the stages of mitosis we'll talk about today, that kind of stuff. So it's I've tried to spell it out as detailed as I can in the study guide. I know that the pictures in the study guide aren't the best pictures. PowerPoints and the PDFs are the best. You're not going to see anything you haven't seen already. I'll tell you that. So anyway, it'll be 50 questions. It'll be multiple choice, and you'll have an hour to take it anytime in that 10-day period. Next week's lab, we're going to start into the skeletal system. So we will have lab next week, but the test is online. The test starts next Thursday, stays open for 10 days. And then we'll be starting new content. So don't worry, the bone test isn't until about 4 weeks later. So we have plenty of time to get ready for that. And you'll need it. You'll need that time to memorize all the parts of the bones and all the bones themselves. Remember, there are 206 bones, and that's not counting the openings and the processes, the bumps, the ridges, the grooves, the flat areas on there. Not only that, but where are they all located, type of stuff. So there's a lot to learn on that. This has got some very practical applications for you. If you have any questions about the test, feel free to ask. I will try to answer it for you.
Sorry, I just had a question about the test. We don't have practice tests, do we? We just have our PowerPoint presentations in this class?
Correct. There are no practice tests in lab. It would be, there's only so many ways to show the same image, and it would end up looking just like the test. And that, well, that might be to your advantage. It's probably not the best way to test.
Hey, I showed up kind of late. What day is the test?
Test opens up next Thursday. It stays open for 10 days.
Okay.
So whatever fits your schedule, whenever you want to take it in that 10-day period, and we're going to go on to new material. So we're going to go on and start looking at bones. But anytime in that 10-day period, when you're ready to take it, go ahead and take it. You get one shot, so you need to do well. I'm sure you will. Okay, any other questions? Okay, in that case, I'm going to open this up and start. Hopefully this is the right one. Okay, I think it is. So we're going to take a look at the first part of this. We're going to take a look at the cells, the cell cycle, and something called mitosis. You've probably heard of mitosis at some point in the distant past or the early past. Cells tend to run in a cycle. They have a life cycle, and the end of their life cycle is usually, in most cells, mitosis. Now, we probably learned that mitosis has the stages interphase, prophase, metaphase, anaphase, and telophase. But today you're going to see that Interphase is not really part of mitosis. We know that the cell is the smallest unit of life. There's nothing smaller than a cell that's alive. A cell can sustain itself and be alive, but nothing smaller than a cell can be alive. Living cells only come from other living cells. There is no way to create artificial living cells. There's no such thing as proto-life in a bottle that you can pour out into a beaker and all of a sudden you're going to have living cells. Living cells come from living cells. Now, the cells in our body, we have billions of cells. We have a variety of types. You saw a lot of those in the last week, looking at the tissue types. The cells are all different, have all different shapes and sizes. They are designed to do certain jobs, like simple columnar cells line our small intestine. They are designed to absorb nutrients. They slide across the membrane and slide into the blood vessels that line the bottom of these cells. Nerve cells are designed to transmit information, and they send it one way and one way only. Blood cells are designed to carry oxygen or fight infection. The cell sizes are going to be variable. Some cells are very, very tiny, like simple squamous epithelial cells that make up our lungs. They are one cell layer thick. They have the consistency of single ply wet toilet paper, and that's probably a gross exaggeration of how thick they are, because they're even thinner than that. Some cells can be 3 or 4 feet long. The longest cells in our bodies are the neurons that show up in our large nerves outside the brain and spinal cord. There's a very large nerve leaving the area of our hip called the sciatic nerve, and it goes from our hip all the way down to our big toe. It runs behind our knee. It is about the size of big around as our thumb, and it's made up of thousands and thousands and thousands of individual neurons, individual nerve cells. That's the sciatic nerve. And it is a very large nerve. And it's the longest cell in our body. It can be 3 or 4, depending on your stature, it can be 3 or 4 feet long. Simple columnar cells, just like simple squamous, very, very tiny cells, but they're all doing a job. They all have the same basic type structures. Now, this is a cell, more or less. But nearly every cell in our bodies have the same basic structure. They have a cell membrane, which surrounds the cytoplasm, which is the goop inside the cell membrane, the goop inside the cell. I've heard it described as cell sap, and within that you have the nucleus, and the nucleus is where we contain the DNA. So every cell, except for one major exception, except twice, has all 3 parts. The nucleus, which is the control center of the cell where our DNA is located. The cell membrane, which is the outer barrier of the cell to protect us from the outside environment. And then the cytoplasm, where all the structures, the internal structures are embedded. Those little things called organelles that function like little organs. They're not living cells, but they're structures within the cell. Every cell except red blood cells has a nucleus and all these organelles. Red blood cells don't. They kick their nucleus out right before their development is finished in the bone marrow, and they get rid of they have none of the organelles. All a red blood cell is a big sac of hemoglobin. That's what a red blood cell is. It's a big sac containing hemoglobin, which carries oxygen. Red blood cells last for about 4 months. They don't reproduce. We make them again and again and again in our bone marrow. They're the only cells in our bodies that don't have a nucleus. So, but they did when they were developing in our bone marrow, but they kick it out. So you could argue it both ways. Anyway, this is a typical cell. We see the cytoplasm is the stuff, the contents in here, these various organelles. This green structure is called the Golgi Apparatus. That's where we package up proteins that we've made. The proteins get made in this these blue tubes here called the endoplasmic reticulum. Don't worry about that for the test. We're going to talk about that in greater detail in lecture. The endoplasmic reticulum surrounds the nucleus. These orange things here are called the mitochondria. They are the power center of the cell. That's where carbohydrates are broken down to release energy, or in the mitochondria. Something we don't see in every cell also are these folds here at the end. Those are called microvilli and villi. Cells will project their cytoplasm out to extend their surface area, particularly cells involved in absorbing things like simple columnar cells can absorb nutrients in the small intestine. So these villi are, it's Latin for fingers. The villi extend off the cytoplasm, and microvilli then extend off the villi, so that we can have a very large surface area available for absorbing nutrients. The small intestine is about 20 feet long. It's about the size of diameter of a small garden hose. And it's lined with these simple columnar cells. If you took every cell lining the small intestine and stretched them out flat, the total absorptive area in that 20-foot tube that's less than an inch across would cover a tennis court. So it's a huge area for absorbing nutrients, even though it's all contained in this little tube. And it's the villi and the microvilli folds that allow this surface area to expand like that. The nucleus is the largest of the organelles. It's the dominant structure. When you looked at the slides last week of the cheek cells, even the cheek cells had a nucleus. All the cells that we saw last week in the tissues had a nucleus. Nucleus is where the DNA is located. The deoxyribonucleic acid is our genetic information. It is, the DNA consists of the strands of DNA and strands of protein called histones that are wrapped together to form thin strands called chromatin. And so when we look at a nucleus, we don't see the DNA. We don't even see the chromatin because we're talking microscopically thin strands here. The actual chromosomes don't appear until the chromatin condenses. And that usually occurs right before cell mitosis occurs. Now, the DNA in every cell is identical. Every cell in our body has the same DNA. The cheek cell, the liver cell, the skin cell, the simple cuboidal cells, the epithelial, the stratified squamous epithelial, the pseudostratified, simple columnar ciliated cells, all the DNA is identical. It's just turned off. The DNA that is allowing them to be a pseudostratified ciliated columnar epithelial cell is turned on. Everything else is turned off. All the information about brain cells and muscle cells and nerve cells and bone cells and blood cells is there, but it's not turned on. All the DNA is there. We don't filter the DNA out and only give skin cells what they need and liver cells what they need. We give them the whole DNA, but it's not active. But it's there. So now, as I said, the only time that chromosomes become visible is when a cell is getting ready to undergo mitosis. Cells live according to a cell cycle. There are two phases of the cell cycle. The extremely short phase of the cell cycle is called mitosis. Now, at some point in our background, we've probably heard that mitosis is cell division. But all mitosis is replication of the DNA. Because you can have mitosis, you can have replication of the DNA, copy our DNA and move it to opposite sides of a very large cell. If there is no cell division, you don't have two new cells. So mitosis is not cell division. It is nuclear replication, nuclear copying. On an exam, if you were to see a question that said mitosis is, and if you marked cell division, you'd be wrong, because it's not cell division, because mitosis has nothing to do with the cells separating into two daughter cells. So don't fall into that trap. It's an easy trap, but mitosis is nuclear replication. The whole process of mitosis is to copy the DNA and produce two sets of DNA in two separate structures. There's no guarantee that the cells are going to divide. Now, the rest of the life cycle of a cell is called interphase. 99.99% of the cell's life cycle is spent in interphase. This is when a cell is busy doing cell stuff, producing proteins, absorbing nutrients, producing vitamin D after being exposed to ultraviolet radiation, sending nerve impulses, fighting off bacteria. This is what a cell does. When a cell is in interphase, it's doing all the cell things that it's supposed to do. And interphase is not part of mitosis. The only thing about mitosis that involves interphase is that the cell will copy its DNA. The cell will copy its DNA during interphase. And even then, the copy may not have been correct. So it has to try and fix it. And if the DNA has been damaged, it won't copy it. And if it's been damaged badly, the cell will commit suicide and destroy itself. So this is what a cell looks like in interphase. Big nucleus, cytoplasm, cell membrane. We can't see much here, but this is when a cell is busy being a cell. It does all the cell things it's supposed to do. There's its nucleus right there in the center of the circle, underneath the label interphase. There's a cell next to it with its nucleus present. Cells are always going through different stages. It's not a uniform, let's all go through interphase and let's all go through mitosis together. It doesn't work that way. Every cell's lifespan and cycle is running on a different schedule. Now, mitosis itself is a very, very quick process. A cell may live for 3 days, or a week, or a month, or a couple of years, or maybe a lifetime. Some white blood cells will last a lifetime because they're part of our immune response. Other cells will only last for a few days. The white blood cells that show up at the site of an infection are going to show up quickly, and then they're going to do their job to kill bacteria, and then they're going to have to break down and die. But some cells will spend a very long time in interphase. The amount of time spent in mitosis is very brief. Now, these are some real-time videos here showing you how fast the cell goes through mitosis and cell division. The process of cell division is known as cytokinesis. That's the cyto for cell, kinesis for cutting in half. They're separating. And that's what we're doing. We are separating the cell into two parts. And so here we're starting out You can see there are the chromosomes moving apart. You can see just how quickly daughter cells form. So the length of time spent in mitosis is very short. If you have even a cell that is only going to live for 24 hours, the time frame for mitosis after that, at the end of the cell's life cycle, will be in seconds. Just a very few seconds to go from interphase to mitosis to 2 new daughter cells. It doesn't take any time at all. So the stages of mitosis, which is nuclear replication, the 4 stages are prophase, metaphase, anaphase, and telophase. If the illustrations in the study guide for mitosis are not clear, the PowerPoints are the best that we have available for those pictures. So here's prophase. In prophase, the chromatin, the combination of the protein histone and DNA start to condense. They start to condense and they produce chromosomes. The chromosomes consist of 2 parts called chromatids. We actually call them sister chromatids. They're held together by something called a centromere. Now, the reason that the chromosomes condense in mitosis is because these, when they're unwound as threads, they are extraordinarily fragile. They're very, very thin. And we're going to sort of abuse the chromosomes a little bit as we push them and shove them around in the cytoplasm and get them to line up on the middle of the cell and then get pulled and pushed apart to opposite ends of the cell. So to protect the DNA, we condense it so we don't have, it's a whole lot easier to protect the DNA when you have all this protein wrapped around the DNA itself and it's all condensed together. It's a whole lot easier to move that than to try and move these individual tiny thin threads of the DNA. So what will happen is this chromatin will start coiling up and condensing and produce chromosomes. The chromosomes are then visible. Up until now, the chromosomes haven't been visible. We also start producing spindle fibers. You may have heard the term spindle fibers in the past. Those are those little fibers that stretch out across the cell membrane that give us those little lines that we see. There are structures in the cytoplasm called centrioles. They look like little barrels. And they will, every cell has 2 centrioles, except the red cells, of course. And right during the buildup to mitosis, when we copy our DNA, we'll copy our centrioles. The centrioles migrate to both ends of the cell, and from there they start sending out the fibers, what we call the spindle fibers. This is all still in prophase. The nuclear membrane, what we call the nuclear envelope, the membrane, instead of the cell membrane, we have the membrane of the nucleus. It disappears, it breaks down. And a structure called the nucleus, which is a dark staining structure in the middle of the nucleus, starts to break down too. Here we see, here's a cell in prophase right here. You can see the chromosomes are becoming visible. That's the dark lines there. We can see the chromosomes becoming visible. We don't see a nucleus anymore. Nucleus is gone. The nucleolus, a darker staining spot, maybe that structure there, we can't tell. No membrane around the nucleus. The chromosomes are starting, the chromatin has condensed. The chromosomes are starting to form with their sister chromatids. This is the first phase of mitosis. The next phase is metaphase. Nucleolus is gone. Nothing remains visible from the nucleus or the nucleolus, so that's completely gone. The chromosomes have fully formed, they've fully condensed, and they line up on the middle of the cell. We call it the equatorial plate, on the line dividing the cell in half. And they line up there so we can make sure that when we distribute the cells, and distribute the chromosomes, then we're going to have an equal number going to both sides. Because we have, in a human, we have 23 pairs of chromosomes. And we've just copied all that DNA during interphase in anticipation of mitosis. In the last few minutes before mitosis starts, we've copied the DNA and made sure it's perfect. And if it's perfect, if it's not perfect, we fix it. If we don't fix it, then the cell stops. It may initiate apoptosis where it lyses itself. We call it cell suicide, or it may try to fix it. And if it can fix it, great, it'll pick up where it stopped. Cells are really picky about this. They want the DNA to be perfect. And so we copy the DNA and we've condensed it. And now we're pushing it towards the middle of the cell to make sure that we get an equal distribution of the DNA to both sides. The spindle fibers have attached themselves to the chromosomes. And they're going to start pulling the chromosomes from one side of the cell to the other side. They're lined up in the middle. Some are going to get pulled to the left, and the others some are going to get pulled to the right. Other spindle fibers that don't touch anything are going to be pushing and shoving the cytoplasm apart. As you know, the cells tend to get elongated when they stretch out, when they're going through mitosis. And it's because the cytoplasm is also being pushed and shoved. Because we want to make sure we have an equal distribution of all the organelles too. Because the organelles the cell's going to need mitochondria and Golgi apparatus, and Endoplasmic Reticulum and lysosomes and peroxisomes. It's going to need all these different organelles. And we want to make sure they get their share of them because we've copied them too. Anaphase is the shortest of the phases. From the time that the cell lines up on the metaphase plate in the middle of the cell during metaphase to the end of anaphase is the shortest of all phases. It occurs very rapidly. The chromosomes are pulled apart quickly. The centromeres that hold the sister chromatids together separate. The spindle fibers are pulling the chromosomes apart. The cytoplasm is being pushed and shoved. The spindles keep pushing and pushing and pushing. And we end up with each pair of chromosomes, one pair on one side, one pair on the other side, so that we end up with 23 pairs on each side. This is what anaphase looks like. You can see the spindle fibers in here where the cytoplasm is being pushed and shoved, and we pulled those chromosomes to opposite ends. During this stage, cytokinesis has started. Cytokinesis, the physical separation into two daughter cells, starts here. It starts with what we call a band of contractile protein forms around the outside of the cell. That contractile protein is the very same protein we use in our skeletal muscles. It's called actin, and it constricts. That's what it does. It constricts. And so it forms around the outside of the cell in the middle and starts beginning to constrict and squeezes a little. It constricts more and more. It squeezes. So I often think if you imagine one of those balloon animals you see at the fair, at the carnival or whatever, you know, that shaped like a dog or something. You know how they can twist the balloon and you end up with, looks like two separate segments as you twist it and twist it. The cell membrane sort of looks like that, only with the balloon animal you can't pull it apart. But here it actually physically does enable it to separate. So anyway, cytokinesis starts in anaphase.
Excuse me.
Go ahead.
Are we going to need to know the 4 stages of mitosis inside now to just be able to identify them on the test?
Just be able to identify them. That's all. Yeah, we'll have a greater detail, greater discussion on this in lecture. This is strictly, this is the being able to identify the stages, So, a telophase, we have cytokinesis continuing. In fact, cytokinesis will run all the way through it. If it's working, it will divide the cell in half. We can see, two distinct daughter cells here. Here we see, it's working on it. During telophase, the chromosomes have reached the opposite ends of the cell. They begin to unwind. Pretty soon they've unwound completely. A nuclear membrane on each one of them reforms. A nucleus reappears, and pretty soon you have two brand new cells, daughter cells, that are now physically separated, or practically physically separated here. Cytokinesis ends in telophase, and we see cytokinesis. Here we have two cells, two brand new cells, and their life cycle now starts all over again. And however long the life cycle is, they're going to, unless something happens to them, they're going to live their life cycle, and then they're going to undergo mitosis again. And you can see where the pinching off has formed two distinct cells. We have a nucleus there and a nucleus there. We can still see the remnants of the spindle fibers, but they're going to disappear. And this will go on. It's generally thought that individual cells can replicate, and if they're going to replicate, they can replicate about 50 times before they stop. We have so many cells in our bodies that we don't notice. We don't, cells wear out all the time, and we're always replacing them. We have billions of cells. So some cells never go through mitosis. Muscle cells never go through mitosis. The muscle cells we have now are the ones we were born with, and they're the ones we're going to die with. We don't generate new muscle cells. They never get to mitosis. Same with nerve cells. Nerve cells don't regenerate. We don't grow new nerve cells. We can repair some of our nerve cells, but we don't grow new ones. So they never get out of interphase. They never get into mitosis. Just about all the other cells in our bodies can replicate themselves. Okay, now let's move on. Second half of this deals with transport across the membrane and the permeability of the membrane. Okay, so cells in our body are covered with water. And where did this water come from? Well, it came from plasma. Our blood vessels are very, very porous. They're not this perfectly sealed tube that we imagine, like rubber tubing or plastic tubing or something like that. It doesn't work that way. Our blood vessels, particularly our capillaries, the tiniest of the vessels, are very porous. They're very leaky. And plasma is leaking out of the vessels into the spaces around the tissues all the time. And we call this interstitial fluid. It was plasma, but it leaks out. Now it's interstitial fluid because interstitial means around the cells. And when it leaks out, it takes out, along with it, hormones and salts and sugars and other nutrients. And so all that bathes the cells, and material moves from the interstitial fluid. It's come out of the blood in the interstitial fluid, and then it can make its way into the cell proper. Even from the capillary, it leaks out of the capillary into the interstitial fluid, bathes the cell, and then gets recovered. Interstitial fluid doesn't stick around long. It gets recovered by our parallel circulatory system called the lymphatic system. The lymphatic system consists of capillaries and veins that recover this interstitial fluid. When it moves into the lymphatic vessel, it's now called lymph. It's then passed through lymph nodes. We've all heard of lymph nodes. We have lymph nodes under our, under our, at our neck, under our armpits, in the groin. Lymph nodes are where white cells tend to hang out and look for things to kill. White cells are the infection fighters, and so they're busy looking for pathogens, bacteria, viruses, things like that, funguses, dead and dying cells, anything out of the ordinary. It is a very strict environment. A cell steps out of line, odds are that a white cell is going to kill it if it's acting in an irregular manner. But that's what the lymph nodes do for us. That's where the interstitial fluid goes into the lymphatic vessels. It gets filtered and becomes lymph. It goes through the lymph nodes and it ends up going back to our blood and becoming plasma again. And it goes over and does this again and again and again. It's sort of like real estate. It's all about location. It's plasma, it's interstitial fluid, it's lymph, and it's plasma again, over and over again. Cells will take nutrients out of the interstitial fluid, oxygen, carbon dioxide, glucose. It will take ions in at once, sodium and potassium, and anything else it needs. It will take what it needs. It will then release waste products into the fluid, which is then absorbed, goes through the lymph system and is then absorbed into the blood to be taken out for filtering and processing. Cells love to do things passively. Cells hate to spend any energy to get, to access the material it needs. Now it will spend energy, but if it can get it passively, it would prefer that. So cells have two ways of getting things, either for passive transport, which is free, or active transport, which costs energy. So cell membrane is made of fat molecules. We call it a lipid bilayer. It's a double layer of fat cells with fatty. It has these little fatty acid tails, and so it is a fat-based membrane. It is, anything that is fat soluble will slide through that membrane. Anything that's water soluble will not get through. It does not. It's hydrophobic. It doesn't let water in. Now there are ways to get through that membrane. The membrane is what we call selectively permeable. Some things get in, other things do not get in. So we have a process known as diffusion that gets us into the cell. Diffusion is movement from a high concentration to a low concentration. Substances move from their high concentration down the concentration gradient to a low concentration until they reach equilibrium. If I were to, and used to do this when we were on the ground, and someday we'll be back on the ground, but I would get a bottle of very inexpensive cologne, a Walmart special, and I would pour a little of that on the floor in the corner of the lab next to the drain. And it would be some flowery odor or whatever. Now, diffusion through the air allows that odor to spread across the classroom very quickly. Diffusion is, the concentration of chemical, the odorant, is very high right where I poured it out. And it's very low in the back corners of the classroom, in the lab. However, over time, the molecules of the chemical, the odorant, the cologne, if you will, if you can call it that, will diffuse. They will migrate passively from the high concentration where it was initially poured out until it spread evenly throughout the classroom, until equilibrium is reached. That is simple diffusion, movement of a substance from a high concentration to a low concentration until equilibrium is reached, until it's equally distributed. It also works as a good example of how we become desensitized to odors, because after a few minutes of smelling the, eau de rose or whatever, or eau de skunk or whatever the odor is, we no longer smell it until somebody comes into the goes out of the classroom, comes back in, and hits them in the face again. So diffusion across the membrane works too, from a high concentration to a low concentration. It works. The only problem is that if it's water soluble, it's not going to get through. Now, gases get through very easily. Oxygen and carbon dioxide, oxygen's always higher outside the cell than inside. And so oxygen will always diffuse into a cell. Oxygen always has a higher concentration outside the cell than inside. It will always come into the cell. Carbon dioxide is always higher inside the cell than outside, and it will always diffuse out of the cell. It moves right across there passively. Anything that's fat soluble will diffuse easily. All of the steroid-based hormones, all hormones based off of cholesterol, that would be all the sex hormones and all of the adrenal cortex hormones are going to be fat soluble. Thyroid hormone is fat soluble and will slide across the membrane. And so with no trouble whatsoever. Other molecules need help. This is simple diffusion. This is what's known as facilitated diffusion, where we have to use some sort of special carrier or channel. If we want water to come into a cell, we have to use a special channel called an aquaporin. It's a tube, lets water come into the cell. It's easy. Water comes in through that tube and bypasses, it's a hole through the membrane, and water gets to bypass the fat-soluble, water-hating membrane itself. It just comes through that opening, like an opening line there. Or perhaps it's glucose. Glucose is a big molecule. Glucose is water soluble. Glucose doesn't have a chance to get into the cell. Now, we all know we need glucose for energy. It's our sugar that we use for energy. We have special carrier proteins designed just to carry glucose across the membrane. It's still free. We call this facilitated diffusion. Whenever we have some sort of assistance that we use to get a molecule across the membrane from one side to the other, doesn't matter which way it's going. If it's going from its high concentration to its low concentration, it can be high on the inside and low on the outside. It can be high outside and low inside. If it still needs help to get across, that's simply facilitated diffusion. Still passive. The only one that costs us energy is active transport. The cell has to release energy using a molecule known as ATP. We get our energy from glucose, but we use ATP as the means to carry energy around for us. In active transport, we can't depend on movement from a high concentration to a low concentration. Here, with active transport, we go from a low concentration to a high concentration. We go against the gradient. We're essentially pumping it and pumping it uphill to get it into the cell or to get it out of the cell. So we work hard to get something either into the cell or out of the cell that we either need or we don't need. We have to go against its gradient to get it in or out. We'll talk a lot about that in lecture. So these are the three ways that we get stuff into or out of a cell. Passive is either simple diffusion or facilitated diffusion. Active, we have to use active transport. Now, A couple terms, we talked about this in lecture already. Solutions, a homogeneous mixture, usually water and a solute. You know, water is the solvent. The solvents are the dissolving medium in our bodies. The solvent is nearly always water. The solute is what's getting dissolved. It can be salts, sodium or potassium or calcium or chlorine or magnesium salts. It can be sugars that are in solution. It can be hormones. It can be other proteins. It can be anything that's in solution. So the solvent and the solute. And when we have a solution, the solution can either be inside the cell, which is the intracellular fluid, inside the cytoplasm, inside the nucleoplasm. So it's either inside, it's the cytoplasm, or the cytosol is the fluid inside the cell. The fluid inside the cell balances the fluid that's outside the cell. Water content inside the cell is equivalent to water content outside, and the same concentrations of particles of solutes are found inside and outside. The cells stay balanced. The intracellular fluid matches the interstitial fluid. We don't want to see movement of water in here, because we'll talk about this, in fluid and electrolytes in AP2, we're going to talk all about all the things that can go wrong in a cell with water movement. Now, the membrane is selectively permeable. Plasma membranes are very, very picky. The plasma membrane of a cell will only let in what it wants. It will only let out what it wants. Lots of things can't get across the membrane. Great big hormones, some of the larger hormones can't get in. They have to communicate to the cell through what we call second messengers. The permeability of a cell determines all sorts of things. To determine overhydration and dehydration. It's going to determine movement of sugars across the membrane. Insulin is responsible for stimulating the cells to make carrier proteins to let glucose come into the cell. If we don't have insulin, or we don't have the right insulin, we're not going to make the carrier proteins, and glucose doesn't get in. And so when we consider diabetes, diabetes is based on either not producing the insulin or not producing the correct insulin. And in either case, without insulin or without the correct insulin, we don't make the proteins, the carrier proteins that allow glucose to come into the cell, and the cells starve. Example here of passive transport. And passive transport isn't just in a liquid. It can happen in the air. It can happen in the gel. I told you about the example of the cologne in the air. That's passive transport through the air. But we can see it through a gel, and that is perfect for us because in our cartilage, absorbing the material, the connective tissue that absorbs shock, particularly in our knee, and lines the ends of our bones so there's no friction here. If cartilage gets damaged, it has to depend on simple diffusion to get its oxygen and its nutrients. It depends on simple diffusion anyway to get its oxygen and nutrients and get rid of its waste products. But when it's damaged, it takes a long time to recover because it is a lot slower through simple diffusion to get its nutrients. Because there are no blood vessels that are in the cartilage. The cartilage is blood vessel free. Depends totally on simple diffusion to get its nutrients and get rid of its waste. Okay, diffusion always moves from a high concentration to a low concentration. down what we call its concentration gradient. If I had a pellet of dye and threw that into a beaker of water, and it was a purple dye, it would be purple, deep purple, right around the site of the pellet. And then it would gradually spread across the entire contents of the beaker until we reached equilibrium. And we would have this purple color, or red or blue or green or whatever it was in the beaker. It would be, in balance. There would be no dark or light spot there. It would all be the same. It would be equal. That's why we call it equilibrium. Diffusion, again, is nothing more than movement down a membrane, down a concentration gradient from a high concentration to a low concentration. It's caused by the random movement of particles, whether it is in air or in water or even in a gel. Air molecules move around, and they're going to push and shove the odorant molecule of the cologne, or the baking bread odor, or whatever it is. So we're going to push stuff across the membrane of the cell. Random movement does this. No energy is involved. Diffusion always works, high concentration or low concentration. The smaller the particle, the easier it is to diffuse. Oxygen diffuses into a cell easily. It is too small to get trapped in the water-hating lipid bilayer of the cell membrane. CO2 is too small to get trapped going out. So we have oxygen and CO2 are two classic examples of simple diffusion across a cell membrane. No assistance needed. Of course, anything that is fat soluble slides through too. Carrier molecules make it happen. If it's something we need and it can't come through, like water, for example, we like to have water in our cells if we need it, or glucose. We like to have glucose in the cells. We definitely need glucose. So, facilitated diffusion or simple diffusion. So again, this summarizes it. Simple diffusion is unassisted. Anything small enough to pass through the membrane without being blocked. Simple diffusion. Now, a variation on simple diffusion is called osmosis. And osmosis deals with the diffusion of water. Osmosis deals with diffusion of water. Now, if the water molecules generally have no trouble getting into a cell through the specialized channels. Water won't come into the cell anywhere on its own, except through these special channels. We call them aquaporins. But in osmosis, the pressure to move water into a cell or out of a cell is based on the concentration of solutes in that water. It's based on the concentration of the solutes in the water. Now, when we say osmosis is movement of water across a membrane from high water to low water, what do I mean by that? Well, if you look at this example here that's in motion for osmosis, you have a beaker, and the beaker is divided in half by a membrane that restricts the movement of sugar. We're dealing with sugar water here. The membrane will not restrict water, but it will restrict sugar. And this is not unlike things we've done in the lab. There's all sorts of membranes that do this for you. So we have a membrane here that restricts the movement of sugar, but not water. On the left side, we have a mixture in here of mostly water with a little sugar, perhaps it's only 10% sugar. So if it's 10% sugar, if the solution is 10% sugar, that means that the percent of water is 90%. 90 and 10 make 100%. So if we have a 10% sugar concentration on the left-hand side, our water concentration is 90% in the solution. On the right-hand side, when we have lots of sugar, our sugar concentration is 40%. It's 10% on the left, it's 40% on the right. I know it's a lot because you see the big green bubbles for the sugar. So on the left-hand side, it's 10% sugar. On the right-hand side, it's 40% sugar. 40% sugar, 40% anything means that the water concentration is going to be 60%. So if we're 10%, if we're 90% water in the left and 60% water in the right, the side of the membrane that has the highest water concentration is this side. Remember, osmosis says it's movement of water across the membrane from high water to low water. And water will move from this side to the right side to try and establish equilibrium. Water will always go from the high water side to the low water side. And that's what osmosis is. It's a passive process. It's a type of diffusion, but it's a passive process, always going from the high water to the low water. If you have a question about that, always look at the percentages of the solution. If it's a 10% sugar solution, that means it's 90% water. If it's a 20% salt solution, it means it's 80% water. The solute concentration will always be given. And so if it's 90% sugar, it's 10% water. If it's 40% sugar, it's 60% water. When you compare the two sides of the membrane, the side that has the greater water number is the high water side. High water means low solute. And so water always moves from the high water side to the low water side. In our bodies, osmosis plays an important role in keeping the interstitial fluid and the intracellular fluid balanced. If a person becomes dehydrated, they lose water from outside the cells. And if the water content drops, our bodies are salty, around 0.9% sodium chloride. If the water content in the interstitial fluid drops because we're dehydrated, the salt concentration goes up, not because we've added more salt, but because there's less water. So if the inside of a cell and the outside of a cell are at 0.9% sodium chloride and we're in equilibrium, everybody's happy. But if the outside of the cell becomes 1% salt instead of 0.9%, and the inside of the cell is still 0.9, water is going to leave that cell and go out into the interstitial spaces, and the cells are going to shrink, and bad things happen when that occurs. We'll talk a lot about that in AP2. Anyway, movement of water across the membrane is driven by the presence of the solute. If the solute numbers are low, water levels are high. Movement across the membrane then is always driven from high water to low water. Doesn't matter which way it's going. And it will always go indiscriminately from the high water side to the low water side, whether it's going from inside the cell to the outside of the cell going in. The example about dehydration, consider this: a person overhydrates. A person says, hey, I can drink a whole case of water in 10 minutes. They can't, but so they sit down with 24 bottles of water and start drinking them, where's that water going to go? Well, it ends up in your blood, and then it becomes plasma, and it leaks out in interstitial spaces. So we flood the interstitial spaces. We lower our salt concentration outside the cells. And when we do that, we lower the salt concentration outside our cells, water goes into the cells, and the cells swell up. And cells hate swelling up as much as they hate shrinking. This is why overhydration is just as dangerous, if not more so, than dehydration. Okay. Now this next one is an example of movement through a different type of medium. This is movement through a gel. Nutrient auger is what we use in microbiology to grow bacteria. So an auger plate is sort of like Jell-O without the flavoring and the coloring agents here. We've taken two separate molecules, one called methylene blue and one called potassium permanganate. And we dropped them on opposite sides of this plate and set back and watch. It's a gel, but the random movement of the molecules in the gel are pushing the potassium permanganate across across the matrix here, across the gel itself, until it's almost halfway across. The methylene blue hasn't moved as fast. Methylene blue is it's a larger molecule than potassium permanganate. It has more molecular weight. And so since it's heavier, it moves slower through the gel. See, simple diffusion like this is based on a couple of things. Particle size is very, very important. Smaller particles move faster and easier than large particles. If you were going to make sweet tea, would you dump a 5-pound bag of sugar in your hot water that you're making a lot of tea? You dump a 5-pound bag of sugar into hot water, or would you drop a 5-pound block of sugar into your water? Okay, which is going to go into solution easier, the 5 pounds of granulated sugar or the block of sugar? Well, the block of sugar is going to take a while. So smaller particles diffuse, go into solution and diffuse easier. Temperature plays a big role too. Now this was done at room temperature. If we had placed this on top of a hot plate and turn it up to where it's a little bit warmer than body temperature, the rate of diffusion would increase rapidly. And this plate would be covered with permanganate, much more rapidly. It will eventually, the brown substance would eventually cover the whole slide. But it takes a couple of days to do that. But if we heated the stuff up, not to the point of melting the auger, but it would increase the rate of diffusion rapidly. Concentration and temperature is very important here. Consider when you're making sweet tea, do you make your sweet tea when the water's hot, or do you make it when the water's cold? Well, you put the sugar in the water's hot because things go into solution easier at higher temperatures. Warmer temperatures increase the rate of diffusion. Now we have a problem in our bodies because we're sort of stuck at 98.6. We can't do much to raise our temperature. But in simple diffusion does better at warmer temperatures. And particle size is important. Smaller particles diffuse faster. Smaller particles diffuse faster. Temperature affects it, and the amount affects it. The more you add of a substance, the faster the rate of diffusion. So, okay. Little experiment we used to do in lab when we were on the ground, we would take bags of dialysis tubing, which is, dialysis treatment filters out waste products. It's like an artificial kidney, if you will. And it is these specialized bags. They're like a type of plastic that's perforated with microscopic openings that are selectively permeable. Some, they're of a set size, and some molecules can get across and some can't. Large molecules can't get across the openings in the bag. We would take a dialysis bag and place, and you tie it off at one end, fill it with 100% distilled water, tie it off the other end, weigh it, place it in a beaker filled with a 10% sodium chloride solution, 10% salt solution, 90% water, 10% salt, 100% water in the bag. Well, if you consider we have a membrane, which side of the membrane has the highest water concentration? Inside the bag. Water will leave the bag and go out into the beaker. How do we know that occurred? Because after 2 hours of running this exercise, we take this is an artificial cell. We take this artificial cell out of the beaker, dry it off, place it on the scales, on the balance beam there, on the balance, and weigh it. And invariably, the artificial cell with 100% water will lose weight as the water has left the cell and gone into the beaker. So osmosis, high water to low water. The highest water is in the bag. Lower water concentration, even at 90% water, is outside. And here we see this is the example. This is what it looks like. It's tied when it's tied off. We have clamps now that we use instead of tying it off. Tying this is really challenging sometimes. So we have a bag with 100% water inside here. The water can pass through the openings in the bag. It doesn't look like it would. It looks like it's a perfectly sealed bag, but it will pass through if it's small enough, and it is. And so the bag shrinks in weight. We can do it, we can reverse the experiment. We can do the same approach and take the bag, a different bag now, and put a 10% salt solution in the bag, which is 90% water, weigh it, place it in a beaker of 100% distilled water. So the higher water concentration is out in the beaker, and the lower water concentration, even at 90%, is inside the artificial cell. Let it set for 2 hours, take it out, dry it off, and place it on the balance. And we discover that the cell has gained water because water moved from the 100% side across the membrane to the 90% side. We create an osmosis experiment using this artificial cell. The solute particles are driving the movement of the water. Solute particles are going to drive the movement of the water. And that is membrane transport. Let me get out of here. And that's, the cell cycle and mitosis and membrane transport. It is challenging to try and present this because we usually do that second part of this is a very hands-on experiment. Usually I set up every table with a couple of those different types of movement across the membrane, and everybody gets their results. And it takes a while to do it, but it is a good exercise here. So I tried the best to get it across. So, any questions on anything? Okay, then lab test 1 will open up next Thursday. It'll stay open for 10 days. Next week's lab, we'll start looking at the skeletal system, looking at bones. I don't know which one I have listed first, if it's the skull or what we call the axial skeleton, but whatever's there is where we'll start. So we'll start, picking up an extra week here to look at the skeletal system is a good idea. So, okay, I'm going to get us out of here, and then I will see you next week. I hope you have a great holiday weekend.